these protein and these protein, but these Rev protein, when it is made, it just works like a working donkey. It carries these messenger RNA and rapidly bring them to the cytoplasm, so that rapidly it should be translated into other protein. So this is also regulating what? Regulating the translation, amplifying the translation by taking the late messenger RNA from the nucleus to the cytoplasm where there are ribosomes, and messenger RNA can be translated into protein formation. Am I clear? Then we come to, virus is not so innocent. We don't allow virus to play these games with us. Even we don't allow our spouses to play games with us. Forget about virus. At least we try. So our immune system tries to fight with virus, but virus has also its defenses. Now let me tell you how. There is a protein which is normally present in our blood. The protein is called ApoBac3G. Very funny name. And it is very naughty looking protein. What is this? ApoBac3G. Right? It has, now this protein is normally present in cells, our cells, and special function of this protein is that if any viral DNA come in our, viral DNA come into our cell, this protein will go and destroy viral DNA. You understand it? Do you think HIV is stupid to allow this protein to work? No. Because if HIV allow this virus, this protein to work, then we will not be having this disease. So actually what happened, this is very dangerous for HIV virus, that ApoBac3G can lead to alteration in genetic material of the virus. We call it, it produces hypermutations, right? There's no need to go into detail that I should tell you that it goes and alters the thiamine and deaminates and produces uracil. I've not told you this, right? You just remember ApoBac3G basically damages the viral DNA. So what the virus should do? Damage this. Am I clear? If someone is coming to damage you, what you will do? No, I think you will run away. But anyway, so because our cell cannot run away from virus, so let's damage it. So actually, this gene produces a special type of cannons. You know cannons? And they destroy this. I don't know how to make cannon, but just imagine this is cannon. Okay? Now, this protein, thank God they didn't put a funny name. In the beginning, they only knew that this protein increases the infectivity of the virus. Why? Because if this protein is made by the virus, then this protein will destroy ApoBac3G, then viral DNA will not be destroyed, more virus will be made and infection will spread. But in the beginning, when they found this protein and gene, they never knew how it worked. So they just call it viral infectivity factor, VIF. So viral infectivity factor produces a proteins, and these proteins destroy ApoBac3G, so viral DNA is protected from hypermutation. Am I clear? This is one defense. There are more defense also. This protein, I will not go into detail because very little known about it. VPR, thank God we don't know much, but it also increases viral replication. It also increases viral replication. VPU protein, this leads to increased viral release. Recently they have discovered the VPU protein, gamma interferon, when it acts on the virally infected cell, produce certain product which will damage the virus. You don't know about interferon. Okay, you will know sometime in your life, right? That when viral infection occurs, interferon is produced in our body, and this interferon enhances certain protein in our cell, and those protein damage the release of virus, don't allow the release of virus. And one of those proteins is tethrine, gamma interferon, lead to formation of protein called tethrine. This tethrine protein does not allow the virus to be released, and this VPU come and destroy the tethrine so that virus can release happily. and go to other cell and damage. So you see, virus is having its own defenses. Number one, by viral VI, come back, TAT, transactivator, replication faster, REV, rapidly bring the messenger RNA out and translate more virus, then protect the virus. WIF, we destroy the apobac 3G so that its own DNA is protected. Then it comes to some product which increase replication also. Then more product, and these products inhibit those natural proteins which inhibit the viral release. So virus can not only multiply very fast, not only multiply very fast, so okay, not only transcribe very fast, not only it will transcribe very fast, it will translate very fast by bringing the messenger RNA, it will protect its DNA, it will release, and it will also destroy the factors which inhibit the release. Natural mechanism. Then NEF. If you forget all of them, please remember the NEF. NEF stands for negative factor. Let me tell you why the NEF is there. Actually, one of the very important antiviral defense we have is that when our cell is loaded with virus, when our cell is loaded with virus, our cells make a molecule which is called MHC class 1 molecule. What molecule? MHC class 1 molecule. This is MHC class 1 molecule. MHC class 1. This is normally produced by our cells. If in your any cell virus enter, you know what that cell will do? That cell will produce MHC class 1 molecule and take the pieces of virus and put over there. What is this piece of virus? Again, it is just like that in your house some enemy come like virus. You make a long what? Flags or long sticks and put the clothes of the enemy on the stick and put it on your roof, so that police which is moving outside the street can recognize there is some enemy inside. In the same way, again, first analogy, if some wrong people enter into your house, right, you want to get them out, police is already searching for them on the road, so you want to communicate to the police that this dangerous group is in your house. One way you can do, take some of their caps, take their, I feel sad, okay, you take their caps and put them on the stick and put it on the top of your house. When police will pass by, they will know that there's enemy inside. So they will destroy your home and destroy you and your, right? Same thing our cells do. When in our cell virus enter, our cells make special sticks. These sticks are called MHC1, and here they put viral peptide, right? And all over the surface of the virally infected cell, class 1 molecule with samples of viral peptide appear. This normally happens in virally infected cells. And then there are special cells which are called T cytotoxic. These cells can recognize the viral protein in association with class 1 molecule. This is the police cell, T cytotoxic, right? These T cytotoxic, when they are moving outside the cell, they can recognize viral peptide with class 1. And if this combination is made, they think, okay, virus is hiding here. So they release certain product and destroy the cell. And if the cell is destroyed, can virus keep its business here? And can virus make millions of copies there? No. So virus is very worried that our DNA, human DNA, it make which protein? Class 1 molecules. Our DNA. So what happened? Virus produces a protein called NEF. And NEF protein goes and disturbs this gene. So this is very sad. Most of the viruses don't treat us like this. This is very cunning, that this virus is present in our cell, and our cell want to report that by taking one piece, but we can only report if we are having which molecule? MHC class 1, and this NEF gene of the virus produces a protein which down-regulates the expression of class 1 molecule. So virus keep on sitting in the cell, and class 1 molecules are not made, so virus is not reported on the surface. T cytotoxics are just desperately moving around, but they don't know where the virus is hidden, and virus just keep on coming out from here. You are understanding? So these are the different regulatory proteins and viral strategy to beat our immune system. Now let me repeat it rapidly. Genome of the virus is not difficult to understand. Most of the retroviral genome has 3 basic genes: GAG, POL, and ENV. GAG is for structural proteins, right? And all of them are structural protein, but GAG is for nucleoproteins, right? Nucleocapsid proteins and matrix proteins. POL is for enzymes: reverse transcriptase, integrase, protease. ENV is for making an envelope. You remember GP120, which bind with CD4 and also can bind with co-receptor with CCR5 on macrophages, CXCR4 on T helper cell, and it also makes GP41, which help in fusion. And then by shifting the reading frames, it can produce regulatory proteins, right? TAT, which increases transactivator and viral, right? And REV, which bring the messenger RNA out, and WIF, which produces a protein which destroy ApoBac3G to protect the DNA. Then VPR, thank God, don't know much. It just increases replication. VPU produces product and inhibit our interferon-induced tethrins, which normally inhibit the release of virus. So when this is neutralized, virus is well released. Then NEF. NEF produces a product which down-regulate the expression of which molecule? MHC class 1. So we cannot report, the cell cannot report the viral peptide in association with class 1 on its membrane, and which cell cannot find the virus? T cytotoxic. Am I clear? Yes, you have a question.
All cells produce MHC class 1?
Yeah, every cell produces MHC class 1 except, I know you are going to ask about how platelets produce. I knew. Her question is that platelet don't have the nucleus. So do class 1 molecules are present on platelet? Answer is yes. The reason being, originally platelets are derived from megakaryocytes. Megakaryocyte had the nucleus, and megakaryocyte nucleus expressed the genes and made class 1 molecule. Then class 1 molecules were expressed on the surface of megakaryocyte, and platelets were just pinched out of that along with class 1. Okay, you got it?
Sir, my question is that it is said that only few cells are antigen presenting cells.
Oh my God, you need a basic correction. Antigen presenting cells are those cells which can express class 2 molecule. Class 1 molecules are expressed by all cells. Is that right? It means antigen presenting cells are like dendritic cell, Langerhans cells, macrophage, Kupffer cells, I don't know, mesangial cells. All these funny antigen presenting cells, like all other cells, express class 1, but specially they express class 2. Am I clear? Okay, that's great. Okay, now let's come back. I think she was trying to confuse me. Okay, so what we were learning? I'm really confused. What was the last thing I was teaching? Yeah, this viral business and genetics. You understand it well? Right? So in the end, to make the things closing here, that I was telling you, what was this duck? Which protease? Viral protease, right? And then in the end what happened, that all these proteins which are made by the virus, right? This is our cell membrane, right? Especially GP41 and what are other proteins? GP120, they are actually inserted in our cell membrane originally, right? And then matrix protein come and attach under it, right? Now virus is being assembled under it. Another thing, in the beginning, there are 2 types of RNA made from this. If the total DNA is transcribed into RNA, that is called genomic RNA. What is it? Genomic RNA. And if partial are done, this is called messenger RNA. From messenger RNA will make polyproteins, and by this action they become functional proteins. In this way, from this proviral DNA, we make the genomic RNA and we make the messenger RNA. Genomic RNA is for the progeny virus, next generation of virus RNA, and this is to be translated into proteins. In this way, proteins are also there and genomic RNA is there. So glycoproteins insert into our membrane, under that matrix protein come, and then protease work and capsids are made, you remember? And these capsids are made around genomic, this is genomic RNA, around genomic RNA, but before making the capsid protein cover, they also put, what is this, restin piece. Is that right? And then this start bulging out, and eventually this virus will escape while taking a piece of our membrane. Eventually virus will be coming out like this, and now these are the glycoproteins. You remember that? Glycoproteins, and under it there should be, yes, rapidly tell me, under it there should be matrix protein, and under that there should be capsid protein P24, and inside that there are 5 people living here, 2 copies of RNA and restin piece, and eventually it will bud off, and in this way virus will be coming out because he has stolen the membrane from us. That is why virus will have our class 2 molecules also, because it downregulated the class 1 but did not downregulated class 2. So our macrophage also produce class 2 molecule. They will be also incorporated here, but I didn't mention because that is not so important at this stage, right? So in this way, one cell can produce millions of new virus, and when more and more virus are going and going, its membrane is being lost. You understand? In the end, cell will die. In next lecture, we will talk about, this was all about the structure of the virus, right? And this was all about that what is the genome of the virus, what is the replication cycle of the virus, and we also talked about clinical points related with it, right? What were the clinical points? I will rapidly repeat, it's worth it. Number 1, we started with GP120. We should know GP120 is attachment. Problem is that co-receptor with CCR5, 3 clinical points. If CCR5 is heterogeneous, homogeneous mutant, infection cannot, HIV can come but cannot produce AIDS. If it is heterogeneous, then slow progression. And if we give a drug Maraviroc, attachment can be slow. Is that right? Then I told you GP120 undergoes mutations, and due to that reason it is difficult to make vaccine. Then I told you, depending upon variation of this, we also have clades. And these clades B is for male homosexual, especially in USA and Western Europe more common. Clade E for female genital tract tropism, and that is more common in Thailand, even though females are found all over the world. Then there is clade C spreading all over the world very rapidly, especially in India, South Africa and Ethiopia. Then I told you about GP120. Clinical point was that it leads to fusion. Which drug inhibit? Maraviroc. GP41, yes, GP41, sorry. Then we came to integrase inhibitor, right? Integrate reverse transcriptase. Thank God you are correcting me. Reverse transcriptase inhibitor. If you apply the pseudo molecules, what was that? Nucleoside, nucleotide, reverse transcriptase inhibitors like didanosine, zidovudine, lamivudine. Or there are drugs which bind with the polymerase and make it dysfunctional. What is the drug? Nevirapine. And then if virus becomes successful to make double-stranded DNA, it will be integrated with the enzyme called integrase, and drug is raltegravir. And then after that, if it successfully makes its structural proteins and it's going to assemble itself, then protease come which will cut the polyproteins, non-functional polyproteins, into functional peptides, and we can use the drugs which can inhibit protease. And what is the name of those drugs? Indinavir, ritonavir, so darunavir, yes, sakinavir, right? And after then I told you about the genes. There are 3 primary genes: GAG, pol and envelope. GAG produces nucleocapsid proteins and matrix protein. Pol produces pool of enzymes and produce envelope proteins. Done. Then there were, okay, they are not here, but there were regulatory genes by reading frame. There were TAT for trans activation. Then there were REV to bring RNA out. Then there was VIF to destroy the ApoBac3G to protect its own DNA from hypermutation. Then there was VPR just for replication enhanced. Then VPU which increases the release because it inhibits the proteins which inhibit the release, right? There was a protein called tetherin. VPU inhibit that, right? So that virus release cannot be inhibited and more efficient. Then there's NEF, and NEF was doing what? Inhibiting the expression of class 1 molecule. So T cytotoxics are not knowing where the factory of virus is working, right? And in the end, I told you that if, yes, very important point which I want to mention. Okay, class dismiss.